The female reproductive tract performs two simultaneous functions: maturing gametes on a continuous cycle and maintaining an environment ready to support an embryo. This complex system operates on a rigorous, non-stop timeline, constantly driven forward by a continuous series of overlapping endocrine signals. Looking at a sectioned ovary, the internal structure is divided into two main areas. The central medulla contains the vascular supply, but the active process occurs in the outer cortex. Here in the cortex, we find the primordial follicles. Each consists of a large primary oocyte surrounded by flat, squamous follicular cells. These oocytes are paused in prophase 1 of meiosis, waiting for a hormonal cue. The cortex operates as a holding zone. Millions of half-finished gametes wait here, completely inert until systemic hormones trigger them to wake up. That trigger is follicle-stimulating hormone, or FSH. When it arrives, the flat squamous cells multiply and thicken into cuboidal granulosa cells, creating a primary follicle. As the primary follicle grows, a thick extracellular layer forms between the oocyte and the surrounding granulosa cells, called the zona pellucida. This glycoprotein ring is a physical barrier containing specific receptors that are required for sperm binding later on. The follicle continues to expand into a secondary, or antral follicle. Granulosa cells secrete fluid that merges into a massive cavity called the antrum, pushing the oocyte into a small mound of cells known as the cumulus oophorus. Outside the follicle, stromal cells differentiate into two layers, a fibrous theca externa and a highly vascular theca interna. You can identify the theca interna cells by their pale, vacuolated appearance, which is a result of their high lipid content. Those lipids are precursors for steroid synthesis. The theca interna produces androstenedione, which diffuses across the basement membrane to the granulosa cells, where the enzyme aromatase converts it into estrogen. The maturing follicle acts as a transient endocrine gland. The estrogen it produces enters the bloodstream to drive systemic changes of the reproductive cycle. The follicular phase climaxes with a massive surge of luteinizing hormone, or LH. This forces the dominant follicle to expel the secondary oocyte, which has now progressed to metaphase 2 of meiosis. After ovulation, the empty follicle collapses into the corpus luteum. The former granulosa cells undergo massive hypertrophy into granulosa lutein cells. Tucked in the folds are darker theca lutein cells. Together, this vascularized structure mass produces progesterone to prepare the downstream uterus. If no embryo implants to provide a sustaining hormone signal, the corpus luteum undergoes apoptosis. Macrophages clear the debris, leaving behind a dense scar of collagen known as the corpus albicans. This highly coordinated sequence can break down, most commonly in polycystic ovary syndrome. In PCOS, excess androgen production halts the cycle. Instead of one dominant follicle reaching ovulation, multiple follicles arrest mid-development, swelling with fluid to form cysts. Because the ovarian follicle drives the endocrine timeline, anovulatory arrest cuts off the hormonal signals required for the rest of the reproductive tract to function. Once ovulated, the oocyte leaves the ovary, gets swept up by the fimbriae, and enters the ampulla of the uterine tube. The wall of the oviduct is built for movement. It features an intricately folded mucosa surrounded by a muscularis made of interwoven circular and longitudinal smooth muscle layers. The mucosa's simple columnar epithelium contains two specialized cell types. Estrogen-driven ciliated cells extend apical cilia, beating rhythmically to sweep the oocyte inward. Interspersed darker, non-ciliated PEG cells secrete nutrient-rich fluid to protect the oocyte and activate sperm. The oviduct operates as an active biochemical conveyor belt, creating the exact microenvironment necessary for gamete survival and interception. Fertilization typically occurs in the ampulla. When capacitated sperm arrive, they release the enzyme hyaluronidase to push through the cells of the corona radiata. Upon reaching the zona pellucida, sperm bind to specific ZP3 receptors. This triggers the acrosome reaction, releasing hydrolytic enzymes that rapidly degrade the glycoprotein mesh. The moment the first sperm penetrates the membrane, the oocyte releases proteases from its cortical granules. This cortical reaction instantly destroys all remaining ZP3 receptors, creating an impenetrable perivitelline barrier that prevents multiple sperm from fertilizing the same egg. If the uterine tube is damaged, this process becomes dangerous. Inflammation from infections, known as salpingitis, can destroy the ciliated mucosa and scar the passage. Without functioning cilia or a clear path, an embryo can implant directly into the thin connective tissue of the oviduct. Because the tube lacks a thick, expandable muscular wall, the growing embryo will quickly cause a life-threatening hemorrhage. The oviduct is engineered for transit. Any delay transforms a fertilized egg into a surgical emergency. Moving down the tract, the embryo reaches the uterine body. The wall here consists of an outer perimetrium, a thick muscular myometrium, and an inner mucosal lining called the endometrium. The endometrium has a stable basal layer against the muscle and a spongy functional layer above containing most uterine glands. Blood supply dictates behavior. The basal layer gets constant flow, while the functional layer relies on coiled spiral arteries requiring progesterone to stay open. Sometimes pieces of this functional layer shed backward through the uterine tubes and implant in the pelvic cavity, a condition known as endometriosis. Because this ectopic tissue retains its blood supply and hormone receptors, it continues to grow and bleed in sync with the monthly ovarian cycle, causing internal scarring and severe pain. The functional layer is designed to act almost like a parasite. If it takes root outside the uterus, it weaponizes the body's own endocrine signals to damage surrounding organs. This chart links the 28-day ovarian and uterine cycles. Rising estrogen drives the proliferative phase, rebuilding the functional layer with straight glands. After ovulation, progesterone initiates the secretory phase, creating tortuous, glycogen-secreting glands. Without an embryo, plunging progesterone constricts spiral arteries, triggering menstrual collapse. The human endometrium is a remarkable transient structure, entirely constructed and demolished every single month under strict endocrine control. Below the uterus, the reproductive tract narrows at the internal os to form the cervical canal, an area specialized for defense. At the squamo-columnar junction, or transformation zone, the simple columnar epithelium of the endocervix abruptly shifts to the thick, stratified squamous epithelium of the exocervix. This zone of high cellular turnover is a frequent site of pathogenesis, making it the primary target for human papillomavirus infections and the origin point for most cervical dysplasia. Continuing into the vagina, the mucosa lacks true glands entirely, relying instead on a robust barrier of stratified squamous, non-keratinized epithelium. Estrogen stimulates these epithelial cells to store glycogen. Commensal lactobacillus bacteria metabolize this glycogen into lactic acid, maintaining a highly acidic shield that inhibits pathogens like Candida. From the highly active ovarian cortex to the protective vaginal mucosa, the female reproductive tract relies on structural determinism. Every microscopic layer is meticulously calibrated by shifting hormones to execute a single continuous mission.